Hello, I'm Livia Zaharia, co-developer of Just-DNA-Lite, and here we want to present you our new library to analyze your own genome individually at home from your own device. Here, since it's a demo, we'll show you the online version that just works on publicly available genome. As you can see, we have here two examples, my own and Anton's. And for each of these, you can just click on import and you'll have it loaded. Obviously, we tried it before, so it's here. Next thing you'll be able to see would be in the input tab here, where you'll see the content of the genome. And we just want to mention that the genomes to be analyzed have to be complete genome sequencing. Usually the ones from 23 and me or Ancestry are not considered to be complete genome. The second tab we're going to look into is the polygenic risk scores. So while we have various traits or disease, each of these are not characterized by only one gene or mutation, but rather by multiple. So we have more than 5,000 PRSs and almost, if we remove the filters, more than 600, almost 700 traits that you can select from. You can just search them by using these wonderful filters. Trait contains, and because I know my family ancestry, let's say thrombosis. Okay, so let's see about deep vein thrombosis. After you select what trait you want to have analyzed, you can just compute PRS and it will be computed and shown here in the PRS results. Since we did try various combination previously, we kind of have a long list, but worry not, we do have these filtering functions as well. So let's see, thrombosis, apply. So we have the latest one we did, deep vein thrombosis, apparently reliable. If we want to have more details, we can see this wonderful bell curve. And here are the values that were assigned. Kind of high, unfortunately for me, and seems that they're consistently elevated and high tail. But since I'm not a bioinformatician, I want a bit more details. And we have this SKA for interpretation, where you can use your favorite model to get some specificity. You get basically the prompt filled in with your results, and let's click it and see what it explains. I have to mention that this video is made in real time, no speed ups, so you, well, we'll see how fast basically you can process stuff. Actually, it appears it is faster than the results from Claude. So in the top 0.1%, so I kind of have high chances. But I have to remind that basically we're checking only the genetic predispositions, and all traits and diseases have also environmental predisposition. So while genetically I'm more likely, I can improve this fact by actually having checkups, by like maintaining a proper diet. So it is not like a direct diagnosis, it's more like a higher likelihood. That's at least in the case of PRSs. In these tabs from above, you can see annotated files, reports, and new analysis. In terms of like doing things, we have this analysis tab where you have various modules where you check basically the gene variations for various causes like coronary, lipid metabolism, longevity, superhuman, VO2 max. Well, obviously you can let them all selected and do like a complete report, but for the sake of demo, I would just want VO2 max because I'm constantly stressed about my low value. You can also have including sample variation annotations. And after you've selected your choice, you just start analysis. And you'll get this analysis running. After the analysis has been computed, you'll get it here. Here we have 4 reports at the moment, and we'll see how fast we'll get to the fifth. And in annotated files, you basically have all the annotation used kind of everywhere, because look, here is the annotated for the genome, and here are files annotated for the variation mentioned in the module. One thing to mention, these modules that you find here are the default that we provide. If you check our other video, you can see how you can introduce your own module based on the paper you've researched or found using AI here. So for example, if you'd find an interesting paper for, I don't know, sarcopenia or whatever, you can just introduce it here to have it created. And it seems that our analysis is complete. If you go to reports, we have the fifth report, and we can click on view here. You can see first from what file it is produced, and let's see. So I have a wonderful net weight of -1. You can see here the gene, the RSID, and everything you'd want, and the positive or negative weight, which should probably explain why my VO2 max was lower than the average female that would take it, even not sports person necessarily. So at least that cleared it up. And another thing to mention actually is that I told you you can run it locally. To do so, here is this yellow button that if you click it, will redirect you to our GitHub page where you can clone the repo and deploy it locally following the instructions in the README files. And if you found our presentation and our deployment useful, please star our repo like you can see here in the right top corner. And follow our channel for more information, more news. Please tell us issues or problems if you have. We're always happy to receive information about what we're doing and how we're helping others. That's all.